Dear students, today we will talk about extraction, purification and the characterization of bioactive natural products. Some part of this extract and this unit is still remaining. We have already talked about extraction, purification and some part of characterization. In the extraction part, we talked about simple methods of extractions like maceration, then soak-slide extraction, then high liquid pressure extraction, and supercritical fluid extraction. So these are simple as well as complex and industrial scale extraction methods. Purification methods like chromatographic techniques and crystallization, precipitation, filtration and many methods of purification including centrifugation we had studied. Coming on to characterization, we had in the last class talked about uh, uh, ab- absorption spectroscopy methods. Simply biomolecular spectroscopy techniques such as spectrophotometer, UV visible spectrophotometer, IR spectroscopy. Then we also studied circular dichroism and similar other techniques. These techniques were for characterization of uh, specific bioactive compounds depending upon their structural nature. Now we in the last class we also talked about uh, detection techniques. In characterization there is separation as well as detection involved. The two techniques that we talked about in the last class were gas chromatography and high performance liquid chromatography or HPLC. In gas chromatography GC, we separate volatile compounds and detect them. In the case of HPLC or high performance liquid chromatography, we separate non volatile compounds and detect them. Today we will talk about the interpretation of the data we get from spectrophotometry and some more techniques of characterization of bioactive natural products. Here we are seeing two pictures. On the left top panel, what we see is uh, a HPLC profile of a purified compound which is an anthocyanin. This data has been taken from a published paper. So in this method, in this profile, what we see at 5.835 retention minutes of retention time is a peak, which is indicative of a singular peak. And before that, whatever peak we are getting at 2.987 and the 3.381 is not clear about its identity. So we will focus on this peak, which is at 5.835 retention time. So in this when we study the spectrum of this particular peak, this only 5.835 minutes retention time peak, when we study its spectrum, on the right top panel what you see is the spectrum of this particular peak. And what we see here is a wavelength scan from 200 to 600 nanometers, and here we see an absorption in the visible range that is around 526, where there is the lambda max of the compound which is represented by this peak on the left top panel HPLC profile at 5.835 minutes of retention time. So on the top right panel, the absorbance in the visible range, that is around 526 nanometers, is indicative of an anthocyanin molecule, which is cyanidin 3 glucoside, but this alone cannot give us the complete information about its identity. So if we look at the other peaks present in this profile, then we see that around 280 there is one peak, and around 260 there is another peak. So these three peaks, one at around 260, another at around 280, and the third one in the visible range around 526 nanometers. All these three combined together give us an indication that the molecule may be anthocyanin molecule, and that too in particular cyanidin 3 glucoside. If this anthocyanin had any acetylation or any acylation, not acetylation, any acylation, then there would have been a peak around above 400 and between 400 and the 500 nanometers. So but in this profile we do not see any peak in between 400 and 450, which means that there is no acylation. So the compound we have got here is an anthocyanin molecule which is non-acylated. So these background informations and some more informations about anthocyanins, which can be understood from their spectrophotometric profile, are given here. For example, any anthocyanin which has no peak around 310 nanometers, if you read the text, which is on the second last para in this on this slide, you will see that no peak around 310 nanometer infers that there is no acylation with hydroxyl or aromatic organic acids like p-coumaric acid and malonic acid. So as I said, the acylation pattern is shown by a peak which is around 310. I think in the previous some time back I said that the peak should be in between 400 and 450 nanometers, but I stand corrected that the peak should appear in between 300 and 350 nanometers, not 400 and 450 nanometers. Similarly, if you see the last para on this slide, only one peak is visible in the visible range of light, which is nanometer, that is 526 nanometers. Hence the anthocyanin should be a monomer. So if there are two peaks, it may mean that the molecule which is indicated by this chemical signature is a dimer, but here it is only one peak, so it is a monomer. And the absorbance ratio of 280 nanometers by the ratio of absorbance at 280 nanometers divided by absorbance at the visible range, the maximum absorbance, that is at 526 nanometers, here will give us an information that the sample is there is no, which means that there is 5 carboxy pyranocyanidin 3-OB glucopyranoside, which is for which the ratio is reported to be 54. But if it is above 54, or it is if it is different from 54, then the 3 o beta 3-O beta glucopyranoside is uh, is uh, uh, is possible if it is near the value 54. So for this compound the value is 56%, which is quite close to 54. Hence this compound can be considered one of 5 carbopyranocyanidin 3-O beta glucopyranoside. So this is essentially the structure shown here in the when you click when you go to the next slide. The structure shown here is cyanidin 3 glucoside, which is nothing but 3-O beta glucopyranoside, whose reported ratio of the maximum absorbance at 280 nanometers and maximum absorbance in the visible range is 54. Now this is all about decoding the molecular signature of an anthocyanin molecule, but anthocyanins are quite well studied. So we have a lot of literature information, and we can always go back to the literature and hunt for the similar or relevant information to identify the compound at hand. But what if we do not have any compound, any background information? How will we proceed to identify a compound without any prior information? So in that case, what we will do is we will study the mass. Remember, I talked about this in the last class also. We use a hyphenated technology called gas chromatography MS, GC-MS, GC-MS. This hyphenated technology in combination gives us the information, the most accurate information of the molecular mass of the compound being studied. Similarly, in that case that was about volatile compounds, but here when the mass spectrometry is coupled with HPLC, then we will be able to study the mass of polar compounds which are non-volatile in nature, which cannot be generally analyzed using a gas chromatography. So when we couple the mass spectrometry with an HPLC, from the HPLC whatever elute, mobile phase elute we get out of that, around 20% enters the mass spectrometry chamber. From in this slide, on this slide, what you see is a pictorial representation of the working mechanism of a mass spectrometry, that is particularly coupled with an HPLC. A mass mass spectrometry which is coupled with HPLC is slightly different in principle and the method of ionization and detection from that of mass spectrometry, which is coupled with a gas chromatography instrument. So let us see how the mass spectrometry coupled with LC or HPLC is working to give us the authentic information about the mass of the compound at hand. So here, if you see on the top left panel, there is one green font text which says analyte or eluent flow. So this eluent or analyte is coming from the LC here, and this LC gives out around 20% of the eluent which is ready to enter the mass spectrometry instrument. Once it enters the mass spectrometry instrument, there is a capillary. You can see in this black font text, it is written spray needle or capillary. So the zero point 0.2 microliter, if the HPLC flow rate is 1 ml per minute, then around 0.2 or let us say 0.2 microliters, that is 200 0.2 ml will enter, that is 200 microliters. So it may be less than 200 microliters also. The maximum accepted volume is 200 microliters to enter this mass spectrometry instrument. So once it enters, the capillary takes up the eluent entering inside it, and through the capillary action it is transported from the entry end to the exit end, which is inside the instrument. At this exit end, there is spraying of the eluent entering this capillary, and this from this spray end, aliquots or droplets of the eluent or the mobile phase carrying the analyte comes out, and these droplets since the inside is maintained at a high temperature, these droplets keeps on shrinking, and when it comes out from the spray needle, these droplets are charged also. When the droplets are charged and because of the high temperature these droplets keep on shrinking, the charge inside and the analyte inside this droplet, which is also charged, and the charge outside the droplets because of the electric field which is prevalent or present here in this chamber, repel each other. The charge on the analyte and the charge which is provided by the electric field present in the chamber repel each other, and at a particular point which is called Rayleigh limit, the charge or the droplet burst and it releases the analyte which is charged and which was present in the droplet into the chamber, and these analytes are guided by electric field, alternating electric field, and it passes through a transfer capillary, and through this again, when it passes through the transfer capillary, each analyte is separated depending upon their mass, and after this, when it passes through a lens or a separator or skimmer, so it will allow no two ions or charged analyte to cross at the same time. Only one will be allowed to cross at the same time, and the charge enters, hits the charge hits the mass spectrometer, which in turn records the impact of hitting it by the charged analyte, and it is converted into a signal, and that signal is displayed on a monitor which is corresponding to the mass of the analyte. So in the lower panel, the spraying of the droplet and how the charging of the droplet happens, and upon desolvation uh, or or shrinking of the droplets, charged droplets in high temperature, that is called desolvation. So the desolvation temperature usually used in these instruments, in particularly mass spectrometry, which is coupled with LC, is around 250 or above 250. In mostly it is 350, 300, 350 degrees Celsius. So it takes very less time to desolvate the droplets, and during that process the droplets shrink to such an extent that it reaches Rayleigh limit. When it reaches Rayleigh limit, there is repulsion between the charge of the analyte and the charge on the droplet, and this is how there is uh, there is bursting of the droplets, leading to release of the analytes. So this is all about the principle which is being used in a mass spectrometry instrument. The uh, the whole of the mass spectrometry chamber is maintained in vacuum, and the vacuum is, of course, to there are many settings which are normally not possible in an atmosphere which is not vacuum. That is why the vacuum condition is required. So let us figure out how many components a mass spectrometry instrument has. The first component here, if you see on the slide, the top panel, there is one capillary. This is the first component. Then there is one ionization chamber or ionization setup, which is of different types. For example, APCI, atmospheric pressure chemical ionization, is one. Then ESI, electro spray ionization, is another. And similarly, MALDI is also another method of ionization for biomolecules like proteins. MALDI is matrix assisted laser desorption ionization technique. So there are different methods of ionization. However, after ionization, when the droplets or when the droplets break, and the ions present inside them, or the analytes which are now charged, enter the portion of transfer capillary, then these ions are separated. So the next important component after ionization chamber is the transfer capillary. After this transfer capillary, where there is separation of the ions, that is only one ion is allowed to pass through this transfer capillary at a time, and then again the ions are again more the ions are further separated, and they are further segregated by using this skimmer. So this skimmer is another component, and then it goes to a mass analyzer. So there are around 4 common mass analyzer systems used in mass spectrometry. One is quadrupole, another one is time of flight, then the third one is a combination of quadrupole and time of flight, that is QTOF, and the fourth one is ion trap. Ion trap has become quite outdated these days. So there is quadrupole, time of flight, Q-TOF. There is 3 methods, and there may be many combination of these, more than one combination. Like in the case of Q-TOF, there is one quadrupole and one TOF. It may be TOF-TOF. There is triple Q, triple quadrupole also. So this is how a mass spectrometry works. The limit of detection of ion trap quadrupole is 1 Dalton, which is the atomic mass unit or the mass of one hydrogen atom. Okay, the spectrum of a compound in a mass spectrometry looks like this. The mass, it is called mass spectrum. For the anthocyanin compound, which we discussed in the previous slide, if we analyze through mass spectrometry, the spectrum we will get is like this. Here there are 2 peaks. The scan has been done in between 225 and 575 m by z. Here m by z means mass, m represents mass, and by z represents charge. So m by mass by charge. Charge is sometimes plus 1 or plus 2, and different charges are there. In the case of GC, this charge is always plus 1, but in the case of LCMS, the charge may vary. So here ideally when we see the charge is like m plus the peak represents m plus h plus species. This species is called precursor iron. A precursor iron of this cyanidin 3 glucoside, which we have already characterized partially using HPLC or UV visible spectrophotometry, is now seen here, and this this product ion is based on our partial characterization. We have arrived at this product ion whose mass may be 449. So there is a product ion which is representative of this compound cyanidin 3 glucoside. Once this compound, this precursor ion is formed, this is m plus h value. This peak indicates m plus h plus. So here after this when it is broken down, the charges depending upon the structure, there are some labile bonds which get broken in the chamber of a mass spectrometry, leading to production of or generation of a product ion. From the precursor ion, there is a product ion which is formed in the inside the mass spectrometry. This is done because of many other techniques. For example, collision induced dissociation is one technique which is used to break down the precursor ion to give out a product ion. This product ion will confirm the structure of the breakdown product, this breakdown products of a precursor ion. This is how not only from this precursor ion which is shown here with m plus h plus mass, now we will be also able to confirm this by the product ion which is obtained from this precursor ion. The product ion here is of 287 M plus H plus mass or M plus M by Z value. So this 287 is a product obtained after deglucosylation or deglycosylation of this cyanidin 3 glucoside. When we remove this glucose moiety which is attached to this molecule, we will get 287 M by Z. Here the importance of the figures after decimal point is somewhat depending upon the instrument which is used. There are now HRMS instruments available, which is high resolution mass spectrometry, where these figures after or digits after the decimal point are very accurately determined to at least 2 places after decimal. But anyway, in the instrument which was used to get this mass spectrum, the figures after the decimal will not make much difference, much meaning. But then here the important thing in a mass spectrum is that every peak has more than one peaks. Actually, no peak is independent here. If you look at the 449 M by Z value peak, there is another 450.63 peak also, which is very much closely associated with this peak. So now we know that organic molecules are made of carbon and carbon, oxygen, hydrogen and nitrogen. Such atoms are used. So in these atoms have their isotopes. So when isotopic masses are considered, there will be somewhat difference in the overall mass of the compound being investigated or being studied. So the peaks which are appearing as shoulder peak of the precursor ion is a peak which is indicative of the compound having radioactive having isotopes of the atoms which are involved in making the compound whose M by Z value is shown by 449.6. So it is very much essential to have more than one peak in mass spectrometry. So every peak should not be as clean as single peak. There should be more than one peak at least, which will account for the isotopes of the atoms present in the compound. So up to this, what we have understood is that the unknown compound, completely unknown compound, can be also studied with the help of a hyphenated technique called LCMS or HPLCMS, wherein HPLC will separate the compound and also detect the compound, and in the MS we will identify the mass of the compound. So the mass, the spectrophotometric profile and its prior studies by using biomolecular spectroscopy, we will get a when we combine all this, when we collect all these data, we will be able to identify the compound. Now if the MS system is not enough to identify a compound. In many cases it may happen, and even if we are sort of 90% sure of the compound's identity, there is another 10% which is needed to be confirmed. So the ultimate confirmation comes with NMR spectroscopy data. So in order to acquire NMR spectra, we need a NMR instrument. NMR is nuclear magnetic resonance, and any NMR instrument measures the nuclear magnetic resonance of the atoms present in the compound. So there are different NMR types like proton NMR, carbon NMR, phosphorus NMR, nitrogen NMR. Different NMR types are there, but the most commonly used and the oldest technique of nuclear magnetic resonance spectroscopy is proton NMR. It can give us a lot of information, and after this only a fraction of information may be confirmed with the help of carbon NMR and then the other types of NMR. So I am not going to explain NMR spectroscopy in detail here, because it is basically used by chemists, and biologists are not used to uh, studying or used to interpreting the NMR data. NMR data comes in spectra, and all from the spectra we will come to know in the spectra carries the signature of the molecule being studied, and each functional group or each region or each electronic arrangement will be wherein hydrogen atom is present will have its signature in the form of a peak. So based on these peaks, we will be able to integrate the functional groups and the electronic density where hydrogen atom is present, and that is how we will come to confirm the structure of the compound after knowing the molecular mass of the compound using the mass spectrometry technique. So here in this particular picture slide, what we see is a NMR spectrum. In this NMR spectrum, we see chemical shift, which is represented by delta in part per million, and there is the hertz, which is the frequency of the NMR which is NMR instrument. NMR instrument gives out a NMR instrument gives out an electric field, or which is recorded as radio frequency. There are NMR instruments. If you go back to the previous slide where there is a drawing of the principle or mechanism of how an NMR spectroscopy works, you will see that there is a radio frequency transmitter. There are different types of radio frequency transmitters used in NMR instruments available today. Among the proton NMRs also 300 500 400 500 600 hertz radio frequency transmitters are used. So with more frequency there will be more accuracy and the clarity of the spectra. So So from this slide what you see is chemical shift, which is how the peaks are integrated or presented. Chemical shift is the shifting in positions of NMR absorptions of the reference and in relation to the sample in relation to the reference, which arise due to shielding or de-shielding of protons by electrons. This can be said in other words like difference in parts per million between the resonance frequency of the observed proton and tetramethylsilane hydrogen. A tetramethylsilane compound is used as a reference, and the sample may be of any type. Samples are usually dissolved in active hydrogen containing solutions. For example, instead of water it is D2O, deuterated water, and instead of methanol there is CD3OD, deuterated methanol. Instead of trifluorocarbon, not trifluorocarbon, okay, let acetic acid, and so everywhere there will be a replacement of the proton by deuterium. So the chemical shift is mathematically equated as frequency of sample minus frequency of reference divided by spectrometer frequency into 10 raised to power 6 ppm. So the spectrometer frequency is 300, 400, 500 hertz or 600 hertz, which is known to us. The frequency of reference is also known to us because trimethylsilane in every solvent will have in each solvent will have a particular chemical shift, and it is finally multiplied with 10 raised power 6 ppm, which is to amplify the signal because the signal is very weak. So now after this we will be able to confirm the, if you go to the next slide where the Spectral Nmr spectrum is shown, we will be able to confirm the structure of the compound. You see in this figure, the if you see the figure caption, it says Proton Nmr spectrum of 4-hydroxy 4-methyl 2-pentanone in bracket diacetone alcohol at 60 megahertz. Assignments of lines, how these peaks are assigned: delta per ppm, chemical shift per ppm 1.23. if you see the delta or chemical shift in ppm here on the x-axis, you will see at 1.23 there is a methyl group, and at 2.16 there is an acetyl group, and then in 2.62 there is one methylene group. At 4.12 there is OH group, and if this compound had in case benzene ring, then it would have come in between 6 and 7. So these signatures are fixed. So based on these signatures we will be able to decode the structure or elucidate the structure. Of course, for an unknown compound, one has to do a lot of hard work to decode the structure, because there may be this is for a simple compound, but not every biological compound is as simple as this. So when there is a complex compound with multiple substitutions and multiple shielding effects working, we will be having a tough time in elucidating the structure. Now you see in the next slide, what you see is proton NMR spectra of cyanidin 3 glucoside in deuterated water at PD 3.9 as a function of concentration. So for different concentration of cyanidin 3 glucoside, there has been a spectra recorded, and all these spectra when shown together, they look like different, and their clarity also differ. So when the concentration increases, the signal intensity also increase, but you will see that sometimes some peaks are separated properly, and sometimes when the concentration is very high, particularly in the case of A, you see 8.6 millimolar. Here the separation of peaks at 7.4 ppm is not sharp enough, and when it is and there is also shift of the peaks, but when the concentration is reduced to 1.4, you see the D, then you will see E, the E here on top of this picture figure. You will see that there is a lot of shift because of the reduced concentration. So a lot of effects can be seen, and it is actually not as easy as it is I have explained. One has to struggle a lot to get to the structure of a compound even after having the NMR spectra. So there are many new additions to this technique. MS can be also combined with NMR, that is not only one MS, two MS in tandem can be combined with NMR. Then just one LC can be also combined with NMR. So all these techniques, as I said, all these identification and separation techniques are sometimes working independently, or sometimes we can use them in combination to get faster and more authentic information. Now there are some other techniques also, like transmission electron microscopy, which can be used to study the structure of compounds, but this technique is not much related to our topic. And X-ray diffraction is very much related, but this is a sophisticated technique, and not many people are able to access X-ray diffraction instrument because it is quite costly, and interpretation of X-ray diffraction data is not that easy. Even the interpretation of NMR data may be easy compared to X-ray diffraction data. So in X-ray diffraction, what happens is that in every compound there is there is electron density map around each atom or each functional group. So Essentially, X-ray diffraction data represents the electron density map of the molecule. So it is to X-ray diffraction gives us direct evidence for the periodic atomic structure of crystals. So how do we study or how do we acquire X-ray diffraction pattern? First, we need to crystallize the compound, and the crystals are kept or mounted uh, at a particular on a particular platform, and then X-ray is shown on the crystal and the diffraction pattern is recorded. X-ray diffraction has been developed to study the structure of all states of matter with any beam, that is in the case of X-ray, with X-ray, and it can study and the beam source may be ions, electrons, neutrons and protons with a wavelength similar to the distance between the atomic or molecular structures of interest. So here the picture shows that when a beam falls on a crystal, it is diffracted, and the degree of diffraction is equal to the distance between the atomic or molecular planes of the compound. So the incident beam is divided into transmitted beam and diffracted beam, and the degree between the diffracted and the transmitted beam is recorded, and which is dependent on the distance between the planes, crystal planes of the molecule. So for an example, let us see uh, in a salt crystal lattice of a salt. A crystal consists of a periodic arrangement of the unit cell into a lattice. So every crystal, when we make crystal out of a solution, it will have periodic arrangement of unit cell into a lattice. The unit cell in turn can contain a single atom or atoms in a fixed arrangement. So the crystals consist of planes of atoms that are spaced a distance d apart, but can be resolved into many atomic planes, each with a different d spacing. Now A, B and C length and the A, B and G angles between A, B and C are lattice constants or parameters which can be determined by XRD or X-ray diffraction patterns. So here there are a lot of physical parameters involved. Now every every compound will not have similar crystal patterns or similar crystals. Some compounds will form cubic crystals, some will form tetragonal, hexagonal, rhombohedral, orthorhombic, monoclinic and triclinic. These are different crystal shapes. Now in a cubic, the physical parameters A is equal to B, B is equal to C, which means that alpha is equal to beta and beta is equal to gamma, and gamma, all of these are equal to 90 degrees. So similarly for other crystal classes also we have different angle and different degrees. So X-ray diffraction can be obtained from powder sample and from crystal sample. The powder sample will be analyzed using a powder X-ray instrument. Here, this is a different type of method, and we for other compounds like crystals, when we have crystals, then we will have this kind of lysozymes diffraction pattern. Here we get to see only dots. All these dots are combined together, and in order to resolve or in order to solve the phases of these dots, we need to have a lot of background information, right from biomolecular spectroscopy data to HPLC data, and their structure, possible structure, their mass, and many other things, functional groups which we record or which we identify with the help of instruments like with infrared spectroscopy instruments. So all these background informations combined together, we get to decode the dots which are obtained with the help of X-ray diffraction pattern. So X-ray diffraction is an advanced technique of confirming the structure of a compound. Beyond this, beyond this, as of now, there is no other technique which can be used for further confirmation of a structure. In the X-ray diffraction also, there are different levels of resolution. For example, in different angstrom, like 2 angstrom resolution, commonly the highest resolution that people have nowadays is of 2 angstrom. When we have 2 angstrom resolution, it means that in any case, when we study covalent bonds, a covalent bond between a carbon atom and a hydrogen atom has around 1.5 or a little above 1.5 angstrom. So if when we say the X-ray has a resolution of around 2 angstrom, we are able to look at the level of one covalent bond. So everything that we see using a, or we record using a X-ray XRD X-ray diffraction crystallography is at the down to the level of one covalent bond. So beyond this, we will not be able to use any other instrument to further confirm the structure. So this is how we characterize a compound. Let me sum up. In order to characterize a compound's structure, we, in my lectures, what I had mentioned were a gas chromatography, a HPLC instrument, and after that a gas chromatography or an HPLC instrument coupled with mass spectrometry. After that, when we are not able to confirm the structure with this kind of hyphenated technologies, we go on to the next level, that is we use NMR spectroscopy to confirm the structure. Even then, if we are not able to confirm the structure, we go for X-ray crystallography. Through the X-ray crystallography data, we will be able to confirm everything down to the level of a covalent bond, that is of around 2 angstrom or slightly less than 2 angstrom bond distance. For today, I will end up with this. I will talk about another topic in the next lecture. Thank you.